Bonjour, bonjour. Donc, en fait, fantastic presentation, Nicolas. And thank you very much, Philip, for the invitation. We've been discussing for a very long time. It's a pleasure to be here and to see everyone at a wonderful institute. So just as a note, like I just realized that the formatting of the slides is off because of the Mac to PC transition. So hopefully this will not cause too many problems, but I can see a few just on the first slide. But anyway, so the title of the talk is Use of Biobank-Scale Genetic Data to Advance Precision Medicine. And to a large extent, it's almost my life story. And I will just start with some very important data. So these are classic experiments that were done just in my neighborhood near in Toronto. And basically what these investigators have done is they've looked how irritable is drug response, or actually in this case, how irritable is pharmacokinetic. And as you can see, these are from, the 60s and 50s actually. And they've given two drugs to twin pairs, identical twins and non-identical twins. And then they have looked at the half-life of the metabolites. As you can see readily, in the identical twins, the pharmacokinetic is extremely similar, whereas in non-identical twin, there's clearly more variation. Now, mind you, these are still twins. These are still non-identical twins, so they do share half of their genome, and yet there's this difference. So we have known for quite some time that drug response, and especially pharmacokinetic, is irritable. But to make this into something useful has proven to be very challenging. And actually, I would argue that except for a few very specific examples, pharmacogenetic has not entered clinical care in a very large way. So indeed, I'm sorry, like the format is awful here, but it's not like this on my computer, I swear. So it leads me to one of our early studies, and this is regarding the antiplatelet agent clopidogrel. So clopidogrel is a prodrug. It is metabolized into the active metabolite through several enzymes, but one of the most important is CYP2C19. And we know that about 40% of the metabolism of clopidogrel, the prodrug into the active metabolite, is through CYP2C19. And then it stands to reason that people with loss of function mutation in CYP2C19, they do not convert the prodrug into the active metabolite at a normal rate, and therefore will have a decreased benefit from clopidogrel. And this has led to an FDA, black box warning of decreased efficacy and suggesting to change agent or increase dose. So we sought to test this in some of the studies we had, and there's some characteristics. So the cure is a study of acute coronary syndrome. But one thing that's important is that these were people on chronic clopidogrel therapy. This was in the randomized control trial. So what we've decided to do is to treat the pharmacogenetic association as we would do any subgroup analysis of a randomized control trial. And essentially what we've seen is that there was no difference in efficacy between carriers and non-carriers of CYP2C19 loss of function mutation. And we've also shown this in a stroke trial, so completely independent. And obviously this came as a surprise. This is not what we're expecting, because I think, biologically, it just makes a whole lot of sense. And finally, these results were largely replicated by my friend Michael Holmes. We've looked at other randomized controlled trial, again using the same design of treating the pharmacogenetic interaction as a subgroup analysis, and could it see an association? And obviously, I'm all into precision medicine. I believe in it. I think it's very important. It's going to become very important. And this left me with a uncomfortable question: is why? So now what I will present is essentially a response to this. And it's, trying to make sense of this, and how can we make progress from this? From knowing that, yes, pharmacokinetic is highly irritable, highly genetic. And yet when it comes to putting this in practice, it's just very difficult. So what I will do is that I will present two new projects in the lab. And the first one is looking at precision medicine really as a gene-environment interaction problem. And the second objective is how to harness large biobank and genetics to evaluate drug effects. And you'll see how they come together. So the first project is to look at gene-environment interaction. And now, why are we, well, first of all, what is gene-environment interaction? Well, it's we can see it as either the environment having a different effect on a phenotype in people with different genotypes, or the flip side is we can see it as people have the genetic variant having a different effect on the phenotype depending on the environment. And they're really like, they're the same, really. But in this case, why the interest in gene-environment interaction is that we have to realize that, pharmacogenetic really is a special case of gene-environment interaction, where actually the environment here is an intervention. It could be pharmacological, it could be others. So I think, to really make progress in pharmacogenetics, then we need to understand gene-environment interaction in general. And the first problem we have is that we actually don't know how important gene-environment interactions are. So this is a classic genetic model. We've got the phenotypic variants, and it can be partitioned into these three categories. There's the genetic variants, so that's heritability. Now on the right side, we've got the environmental variants, and that's environmental with like a uppercase E. So like it's just not the environment, the lifestyle. It can also be like random processes and so forth. And finally, in the middle, we've got this, oddball, which is the variance of a trait that is due to gene-environment interaction. And we do not have good estimate of this. And yes, and yet I would argue that this is central to precision medicine. So there's many reasons why we need to have these estimates, because if we know this, then, it can help us better determine which population would benefit from preventative intervention. Obviously, it would help us with pharmacogenetics. Another thing that we think is very important is nutrigenomics, and I'll come back to this. And ultimately, if we want to personalize intervention based on genetics, we need to know how much of the variance of the outcomes are due to these gene-environment interactions. So we have worked hard to come up with an algorithm to estimate the overall proportion of the variance of a trait that is due to genome-wide interaction variance. And that's the same way that we can estimate heritability without knowing exactly what are the associated variants. We can also do this for gene-environment interaction. Now, I will not go through the details, but I would happy to do so after. But I will just share with you, what we've learned from this exercise. And one thing that we've learned is that actually the most difficult step is step one, is how you clean the data. And there are two things with interactions that are, I think, very specific. The first one is that heteroscedasticity really is key. And heteroscedasticity is when the variance of a trait, varies between groups that you're interested in. And that makes a huge impact on estimation of gene-environment interaction. And the other thing that, sorry? So essentially it's when the variance of a trait is not constant across the factors you're interested in. So I'll just simplify it, but if you were to look at, you're interested at the genetic effect. So we've got, three genotypes, A, B, and BB, okay? And in our analysis, we don't really look at this. We look at the difference in the B between the different genotypes, but there can also be differences in variance between the three genotypes. And in most analysis, that doesn't make a difference. But in these type of analysis, that makes a difference. And where it makes a difference is that if you're interested in gene-environment interaction and the environment says waste-tip ratio, the phenotype has to have a constant variance across the waste-tip ratio range. And otherwise, it creates some statistical bias. It's not the biological bias, it's statistical bias. And it can get very tricky too. So we've spent a lot of time actually just to derive a procedure to adjust for this. which is like a rather mundane problem because that's very far from our goal, but it just makes a big difference. The other thing that we've learned that is counterintuitive is that when it comes to gene-environment interaction, missing values or imputing missing values doesn't bias towards the null. It bias towards type 1 error. And it took us some time because, again, you think it's missing data, we're going to impute it, it goes toward the null. Actually, it's the other way around. So once we found it, we finally, could have good simulations, and we're satisfied that we're getting good estimates. So the next step we had is, okay, let's try to apply this to the UK Biobank. We need very large sample size for this. And in this case, we were interested in looking at interaction with waste tip ratio. So this is the environmental variable that we've chosen to select. The reason we've selected is we think, it's a pretty good, measure of our obesogenic environment and diet and exercise. And we've looked at this with a number of cardiometabolic traits. I skipped through this, but our method is we're dividing the genome into blocks, and then we're looking at each block. These are very large blocks, and then we're combining them together and putting them together. So these are the main results. Okay, so I'll just walk you through these results. So again, this is always interaction with waste tip ratio. So the environment is always waste tip ratio. And on the x-axis are the outcomes. So if you look at the first one, hypolipoprotein A, then in red is the genotype, R squared, so that's the irritability. In green is the waste tip ratio, variance explained. And then in blue is the gene-environment interaction variance. So there's a few conclusions here. The first thing is that, we could recover known irritability for these traits, or estimates agree with what has been published. The other thing that you can see is that gene-environment interaction, or in this case, gene-waste tip ratio interaction, do exist. And we did find significant result for a number of these traits. The strongest was interaction for CRP. So gene-waste tip ratio interaction for CRP was the one with the largest component, explaining 7% of the variance of this trait. And the other observation here is that, generally speaking, the gene-environment interaction component is proportional to the variance explained by waste tip ratio, which I think, makes sense. Now, obviously, our first reflex here is like, what have we done wrong? Okay, so we've done a bunch of sensitivity analysis. The first thing is that we repeated the same analysis, but instead of looking at waste tip ratio, we've looked at a metric of exercise exposure. And again, we did this because we thought it's related to waste tip ratio, but it's there's almost no irritability for exercise exposure. And just to, remove any possibility that the waste tip ratio irritability that explained this. And we found largely consistent result, and we've used another exposure, which is smoking, and there was actually no significant result. And then finally, we use a randomized trait, and then the results were completely null. So with this, we can ask a few questions, and actually we've asked two secondary questions. The first one is, what is the genetic architecture of gene-environment interaction? So this is that's a slide that's a little bit difficult, I think, to understand, but the key point here is that in the upper panel in red, it tells us how much of the irritability of a trait we can recover by selecting SNPs that are either strongly associated with the trait or have strong interactions. And what this shows, and this is known, okay, is that when it comes to irritability, you've got a relatively small group of variants that explain a disproportionate proportion of irritability. And that's illustrated by, these here. So if you pick, SNP with p-value lower than 10 power minus 5, you can, recover up to like 50% of the irritability for most traits. This is not the case for gene-environment interaction. So the genetic architecture of gene-environment interaction seems to be a whole lot more polygenic. There's less of these variants with very strong interaction. Rather, it seems to be spread over many more variants. And I think that this explains why it's been so difficult to identify SNP with gene-environment interaction, individual SNP, because they will individually explain only a little part of this gene interaction variance. And then the second thing we were interested in, which is a good question in it by itself, I also think it's a good validation, is if we incorporate gene-environment interaction into polygenic risk scores, would we improve their predictiveness? And here you can see in this figure, you've got in red, the polygenic risk score without interactions, and then the other lines, which are kind of all squished together, are polygenic risk scores, including the additive genetic component in addition to interaction. And as you can see, there is a slight improvement in the variance explained when you include gene-environment interaction into polygenic risk score. This was done in an independent sample. And it shows, first of all, it validates the model because it shows, yes, these interactions are there because now we can create scores using them that have a better predictiveness. But the other conclusion here is that the improvement was actually marginal. So it's kind of interesting as a proof of concept. Would it make a better CRP genetic risk score? I am not fully convinced. So just to summarize the findings here, so we've developed this method for gene-environment interaction. We've shown that these interactions, they do exist. In this case, for waste-to ratio, they can explain up to 7% of the variance of C-reactive protein. For the other traits, actually, the proportion of variance explained was quite small. And we've also shown that it can lead to an improvement in polygenic risk score, although the improvement is modest. So this is great, but I think, this is not the goal. This is creating a tool, and the tool has another goal, and the goal is precision medicine. Okay. So the first question we thought to address is the one of nutrigenomics, and it's this concept that different people will have different response to diet according to genetics. And there's a whole field on this. There's a whole field of nutrigenetics. And yet, we do not know our outcomes. Do these gene-diet interaction really exist or not? If it ends up that they explain 0% of the variance of traits, then I know this field is invictorable, right? But there's never been this exercise. So what we thought is to quantify gene-diet interactions, and then we could see, well, can we use this to improve genetic prediction of response to diet? Again, we've done this in the UK Biobank. You see the sample size varies because not everyone in the UK Biobank has the same diet information. Some people have more complete information than others, and this is why there's this discrepancy. And here we decided to do a very comprehensive survey. So we look at 31 cardiometabolic outcomes and 23 dietary exposures, including all main diet score that are published and used, as well as individual nutrient components and principal components of diet, and so forth. So again, this is using the method that we developed, and then once we have this, we have a bunch of secondary analysis to try to see, well, are there pathways or can we do scores and so forth. So okay, so these are the main results. So on the x-axis, you've got the different dietary component. On the y-axis are the 31 outcomes that we add. And if you see a dot and a square, that means that there was a significant gene-diet interaction component or variance explained for this particular pair. And, you can see a few patterns. The first one is that, thankfully, there are some significant gene-diet interaction, but they're very rare, okay? And in fact, out of the 730 pairs we've tested, there was 20 that were significant, okay? And that's, that's like 2% or so. The other thing is that the significant gene-diet interaction are concentrated with a few diet exposures. And the one that really stood out is alcohol. Coffee was also there, and there was also something with the Mediterranean diet that we've seen. So with this, then we decided to look at our second question, which is, can we predict response to diet based on genetics? So here I just need to introduce two concepts. Okay. The first one is the gene diet score. So the gene diet score is a polygenic risk score of interaction regression coefficients. Okay. So essentially, it's a weighted sum of genotypes at selected genetic variants. And this gene diet score is calculated strictly based on genetic information. Okay, so at birth, we can calculate this gene diet score the same way we can do a BMI genetic risk score or a CAD genetic risk score. The second, concept is the realized gene diet score. And in this case, we take for each participant, we take the gene diet score, so the polished risk score, and we'll multiply it by the corresponding dietary exposure. And this is the one really that should be associated. So our first analysis here was to look, is the realized gene diet score associated with the corresponding outcome? And as you can see, we had several diet exposure for these scores, so that this is the color coding of these points. And then on the y-axis, we've got the corresponding outcome. And so we've taken all of the diet outcome pairs that had significant gene diet interaction component, and we've derived this gene diet score, and then we've derived the realized gene diet score. And then we look in an independent validation sample, are these realized scores associated with their outcome of interest? And the answer is that for all but one, the association was significant and the direction of effect expected. So if we look at the first one, which was the strongest, so we have seen that there was a very significant gene-alcohol interaction to explain apolipoprotein A as the outcome. And when we do the realized gene diet score in a dependent sample, we can see that this score actually predicts ApoA as it should. So that's a nice, validation. We're quite happy with this. But there's a more intuitive way, I think, to show. There's a more intuitive way to show this. Okay, and in this analysis, what we do, again, this is done in a validation sample, is we take our validation participants, we calculate the gene diet score, so not the realized gene diet score, just the gene diet score, and then we partition the participants according to decile of these four. So the partition of participants is strictly based on genetic information. People would be allocated to this, these decile at birth. And now in each decile, we look at what is the association of alcohol, in this case, with the outcome, okay? And if you look at the one that really stands out is the blue curve, and here in this case, the CRP is the outcome. And what it shows us is that in individuals in the top decile of the score, for every additional alcoholic beverage people drink daily, there's this strong CRP reaction, there's this strong inflammation reaction. But in folks in the last decile, there actually no significant interaction. So strictly based on genetics, we can determine groups of people that respond to alcohol in terms of inflammation from a strong reaction to a rather minor one. And there's a few important points here to mention. So the first thing is that the gene diet score is not associated with diet, by itself. Okay. The other thing is that the gene diet score by itself is not associated with the outcome. It's only the realized gene diet score that's associated with the outcome. And again, these were all done in an independent validation sample. And then we've gone, one step further. So we took the significant score with this and be like, where would they be associated with harder outcomes? And there's really one that stood out, which I think is pretty cool. Actually, there's two. And sorry, the formatting is just terrible. That's the whole translation from Mac to PC. But, basically, what we've seen is that there was an association. If we took the ApoA gene diet score, there was this association with CAD. So people in the first quintile, there was a significant association of alcohol with CAD, but not in the last quintile. And what's really interesting is that we know that alcohol increases HDL and ApoA. That's well known. And basically, it suggests that an individual with a larger response with alcohol and HDL are the ones actually at our increased risk of CAD. And we'll do more work on this, but we're quite intrigued with this. So that actually leads to a marker that we see in the clinic. And the other strong association is with gout. So basically, we know that, alcohol is a classic risk factor for gout. It's been in the textbooks, forever. But basically, we can pinpoint 20% of the population that are particularly susceptible to gout with alcohol. And on the other end of the spectrum, there's this 20% of the people that there doesn't seem to be an association between alcohol consumption and gout. So just to summarize here, so essentially from our nutrigenomic survey, we identify 20 significant interaction pairs that explain from 2 to 20% of the outcome variance. With this, we've derived the gene diet scores, and we've shown that there was significant association, and we think that this can be translated into advice on who's susceptible. Interestingly, the discretionary dietary component are the one with or interactions, especially alcohol. And I think when we think about it, it might make sense because this came later on in mankind's history. And perhaps, we haven't developed all the mechanism to buffer the effect. But one thing that's really important, and that's a theme that, we've seen is that, and other analysis as well that haven't shown, is that when we do these large screen for genome-ramp interaction, actually there's only a tiny proportion of exposure outcome pairs with significant genome-ramp interaction. So if we had taken a random pair, like we say, oh, we think that eating sugar will, interact with genes to explain glucose, that makes a lot of sense. But just from a strict probability point of view, the chances of finding these interactions is very small. In a completely different project, completely different methodology, looking at different traits, we come up with the same proportion of traits having significant genome-ramp interaction. So what we posit is that the right way to do so is to first look at exposure outcome pairs with significant genome-ramp interaction before going further and trying to have, personalized recommendations. So part of this, so the methodological part of this has been published already, and then a lot of the other data are new and hopefully will be submitted. So and now I'll just move to the second part, and is to how to estimate the effect of drugs using genetics in biobank level data. And I'm sorry because you're missing a big part of the slide here. But basically, the goal here is to look at the real world effect of drugs using biobanks. And why do you want to do this? Because right now there's only two ways to do it. One is to do a randomized control trial, and you measure a biomarker before you give the drug, you measure the biomarker after. And this is great, and that's a gold standard, but it's very costly, and it cannot be done at scale, and you cannot explore. You need to do these super costly studies that are meant to just study one trait. The other way to do that is you can take a big epidemiological samples, and then you test this. But there's a big problem with this, and the problem is that there's a lot of biases, okay? So for example, if you take the UK Biobank, okay, and you ask the question, are statins protective of cardiovascular disease in the UK Biobank? Well, you will come up to the surprising conclusion, and believe me, there's a lot of TikTok, influencers that have come to the same conclusion, that taking a statin actually causes heart disease. And the reason is obvious, right? Is because people get a statin because they're at risk of heart disease, right? So the group of people taking a statin are at increased risk of heart disease. Doesn't mean that statin is bad or doesn't prevent heart disease. And it's the same for cholesterol. People with higher cholesterol are the ones that are getting a statin. And the consequence of this is that, you're really underestimating the effect of a statin. If all you have is a cross-sectional slice of society and you know who's taking a statin and you know their cholesterol levels. So what we're proposing to do is to use genetics and more specifically optimization of irritability to try to estimate the real world drug effect. Okay. So we have named this causal irritability mapping, and the software is GenePy. And basically the principle is that a trait's irritability is maximized when it is unaffected by environmental factors such as medication usage. And if we can do this, then we can use cross-sectional data to estimate drug effect. Now, it's actually, it's a bit of a mind twister, like just to understand that it's not easy to explain. But essentially, if we take cholesterol as an example, okay, well, it turns out that people that are at highest genetic risk of having high cholesterol are also the one more likely to take a statin. Okay. So if you take, a population and their cholesterol measures and you calculate the irritability, the usage of statin is really killing the irritability because people that should have the highest cholesterol have the lowest observed cholesterol, and the genetic association will therefore be much weaker, okay? And essentially what we're doing here is that we test different adjustment of statin on cholesterol until we reach a peak of irritability, which would be the irritability if, doctors were not, messing with LDL levels and medication. So, okay, so this is the overall principle. There's a bit of nomenclature here, so I'm going to show a few of these models. So, here, Y is the outcome of interest, I is the intervention, G is genetics, obviously, and then we've got models with U a compounder, it's C a collider, and in the simple model, genetics only impacts the outcome. In a more complicated model, we've simulated cases where both the compounder and collider are also irritable. So just to give you that's an overview of the method. Again, we're dividing the genome in big blocks, and then we calculate the correction for the intervention, and we can use multiple intervention in the same model. And then we've developed a maximum likelihood function to home in the maximum irritability. This is like very computationally intensive, but you use a bunch of tricks, and I will go into details, but rather I will show the results of the simulation we've conducted. Okay. So the first question, and really the reason we developed this, is because of reverse causation. So when we say people with higher cholesterol are the one getting statin, that's reverse causation. Okay. So in all of these results, in red are the results of epidemiological results, and in blue are the results using our method. And as you can see, on the x-axis is the true intervention effect, and on the y-axis is the estimated effect. And the left panel, this is a situation where you've got reverse causation bias. So as you can see, and as expected, the epidemiological associations are quite biased, and especially so when there's no true intervention effect, but you have reverse causation, then you've got a huge bias. And or estimates are non-biased, at least when the effect size is reasonable. At that huge effect size, and you start to have a slight bias. Then we've tested collider bias, and I would say to our surprise, actually, our method is robust to collider bias, whereas epidemiology is not. Okay, that's what's shown. And this is especially important when the true intervention effect is 0. So that's really the type 1 error, right? Which is especially dangerous. Okay. Then we continue with compounder bias. So when there's a compounders, and here we've got actually 4 sets. We've got epidemiological analysis with and without adjustment for the compounder, and CHM analysis with and without adjustment for the compounder. And here in both cases, there is a bias when you're not adjusting, And when you adjust for the compounder, the bias is you can remove the bias from the compounder. So the important conclusion here so far is that whereas epidemiological analysis are susceptible to reverse causation, collider, and confounder, CHM is robust to reverse causation, robust to collider, is susceptible to confounder. Although if the confounder is observed, we can adjust for it and still get unbiased estimate. A particularly interesting situation is when a variable is both a confounder and a collider. And in this case, this is, checkmate in epidemiology. You cannot adjust for it because if you're not adjusting for it, then the confounder is going to be there. But if you adjust for a collider, this is when you create a bias. Okay. So interestingly, with this method, because it's not susceptible to collider bias in this situation, if you adjust for the collider slash confounder, you can get unbiased estimates, is the point here. And, okay, I'll just skip this quickly. Okay, and then the second set of simulation, I'll just go a little bit faster here, is in the more complex model where both the confounder and collider are also irritable. Okay, and maybe I'll go straight to the conclusion here, is that when this happens, this can actually bias our results. But there's a workaround, is that if you remove all the genetic determinants of the collider and confounder, and you only keep the genetic determinants of the outcome, then you can remove this bias, but it comes at a cost. And the cost is that the estimates become less precise because we've removed some of the irritability. So there's a way around this, but it's not a free lunch. And essentially, this applies to, both the reverse causation and the collider and the confounder. Okay. As you can see in these cases, the epidemiological, estimates are really off. Okay. Now, finally, the last set of simulation before we move on to real results is, do we have enough power to conduct these analyses? And here, you have a few important components, and the component, obviously, sample size, but the other important thing is how irritable is the outcome. And essentially what we've shown is that in a UK biobank size study, you need the outcome to have more or less 5% irritability to have enough power to do this. And as you can see, if you've got more irritability, then you get very precise estimate. But, even with UK biobank size sample, now you're in a territory where, there's some analysis you can just not do because there's not enough power. And we'll need even bigger. But the good news is that it's actually realistic, to be done for a lot of outcomes. Okay. So, okay, again, the slide problem. But just to, summarize here some of the key findings. So essentially, there are 3 ways to estimate the impact of a medication on a biomarker outcome. So randomized trial, cross-sectional, or, CHM. Again, cross-sectional and CHM have the advantage that only one measurement of the biomarker is needed. You don't need pre-post. So we can use biobank data. They are robust against confounding, but for observational and CHM, you need to observe the confounder and adjust for it. Obviously, randomized trials is immune to it. Then both randomized control trials and CHM are robust to collider bias and reverse causation and indication bias, and that's not the case for cross-sectional study. So our next step was to test this in the UK Biobank, and the model we've used is lipid therapy. And the reason we choose this is because we know what the true answer is. These trials looking at how much a statin will reduce, LDL cholesterol are there. Plus, these are irritable traits, and there's tons of people on statins and other lipid-lowering agents. So that works really well for us. Okay, and so this is the main results, and basically we look at 9 lipid-lowering medication. Here, what's important is that in the purple dot, the first one is the the estimate of the effect from randomized controlled trials. So this is not from the UK Biobank. This is what is known in proper studies. And people get, the measurement of cholesterol before and after. In light blue is our estimate from CHM. And then in red is the epidemiological estimate. So if you just strip the UK Biobank, you try to estimate this. So what you will see is that in all cases, the CHM estimate are much closer to the randomized control estimate. And actually, there's a trend here for LDL is that the epidemiological estimate are consistently underestimating the effect of the drug. And this is because of this reverse causation thing. It's because people with higher cholesterol to start with are the one are giving a statin. The other thing that's really interesting is that using CHM, we could actually rank the statin from the most potent to the least potent. Okay, and that's not something you can do with other genetic methods. But here, we can actually see that, yes, pravastatin is not as potent as rosuvastatin using it. So not only the direction effect, also the quantity. And the last thing that's interesting here is the triglycerides. So this is the last box on the N. And, so the fibrates are given with people with very high triglycerides. And in this case, the epidemiological association actually goes in the wrong direction of effect. If you didn't know anything about triglycerides and fibrates in the UK Biobank, and you say, are fibrates associated with lower triglyceride, you come to the inverse conclusion. You say, no, triglyceride increase. Fibrates increase triglycerides. And that's because only people with extreme triglycerides get prescribed a fibrate, whereas CHM actually gives a proper effect size estimate that is consistent with RCTs. Okay, and so we have discussed this. And then obviously this is good. And then, we had a quick look at, novel effects. And in this case, we look at waist-up ratio. And basically, we could reproduce known effect of some drugs on waist-up ratio and actually found a new effect of fibrates on waist-up ratio based on this. There is no randomized control evidence we could find of the effect of fibrates on waist-up ratio. There's some model system that have shown that in mice, they do use some adipose tissue. So I think it raises a interesting, hypothesis. So, and we're almost there, but I think there's an important discussion point is how does CHM compares to Mendelian randomization? And I would say that they're kind of like, the two sides of the same coin, because they're quite complementary. So if we look at some of the key differences, so for CHM, we need the outcome to be irritable. Whereas for Mendelian randomization, it's not the outcome that's need to be irritable, it's the intervention, it's the exposure. And that's perhaps really the key difference. The other big difference is that CHM gives a real-world effect estimate. So with Mendelian randomization, you can test that HMG-CoA reductase should be associated with lipids and coronary artery disease, but it won't give you the effect size estimate for a specific drug. Whereas with this will give you effect size estimate for specific drug, and that's what we mean by real world. In Mendelian randomization, you need to select your instruments. You pick your SNP with strong association with a trait. In CHM, because we look at irritability, we actually don't need to know the genetic architecture of the outcome. We just need to know it's irritable. Another big difference is that NMR, it can be done with summary level data, which is super practical, whereas for CHM, you do need individual level data. There's no other way to calculate the irritability. You need individual level data. And I guess I'm missing the other ones because of the slide formatting issue. But I guess we've covered the main ones, and maybe it's gonna come back later. Oh yeah, the last one is pleiotropy. So pleiotropy is, I think, in one way, a pitfall for Go. There's some methods with MR that can handle pleiotropy. In CHM, pleiotropy is when either like the compounder or collider are irritable, and we found, a method to work around this to remove them. So I think in both cases, they're susceptible to pleiotropy, and there's ways to adjust for it, would be the conclusion. Okay, so just to briefly discuss this part, and this was done by my PhD candidate, Ricky Lally, which has been absolutely fantastic. But essentially, it leverages genome-wide irritability to estimate drug effects, more practical than RCT, yet robust to reverse causation, collider bias, compounding bias, but only if we adjust for it. It's still susceptible to residual confounding or unobserved confounding or confounders we observe but we don't include in the model. And then, we validate this with lipid-lowering medication. And obviously, again, this is a means to an end. We're not, the goal here was not to replicate, the effect of lipid-lowering drug, which we know. But what we're excited about then is that we can use this to have subgroups. And then we can say, can we predict in a UK biobank size, study, whereas statins have more efficacy in this subgroup of participant or that subgroup. So this is really what we want to do, is then use this, to generate hypotheses for precision medicine. So as, the overall conclusion, I'm just going back to the first slide. So we know drug response is irritable, just like other traits. Why hasn't this, entered the practice? Well, if we look at the first part and thinking that gene-environment interaction and thus, gene-drug interaction are highly polygenic, I think we need to have a much more polygenic approach to pharmacogenetics and not focus on one variant in CYP2C19, as we have done, among others, but looking at really the scores, as we've shown for the gene-diet interactions. Not to neglect, rare variants, which, I haven't talked. I think another big conclusion here is actually gene environment or gene diet interactions are rare. When we do these huge screens, we screen like hundreds of potential cares of exposure outcome, and only like 2% actually have significant gene environment interaction. Though these are the ones to focus on for precision medicine. But if you pick one randomly, I think the chances are like there's no variance explained by gene environment interaction to start with. And that's not great for genetic-based precision medicine. And finally, I think, to do all this, if you bring the both projects together, we've got this high polygenicity, so you need big sample size and to test these pharmacogenetic hypotheses. And this is why, we want to combine with the other method that enables us to use large, publicly available databases. Ultimately, I really strongly believe we need these smaller, super well phenotype characterized study to really bring the evidence to bring it to the clinic. But as a screen to generate these hypotheses, I think that the large biobank and associated methods are the way to go. So I will thank you all for your attention, and obviously thank, students and others.